Good morning, everyone. It's very difficult to follow very infectious and enthusiastic presentations or talk from colleagues from Spinco and Shimadzu. But it's a great honor to be invited here. And it's very difficult to all say no to Arvind when he calls, Such an young, enthusiastic, energetic entrepreneur and a great colleague to have known him for so many years now. Such a young age, he has achieved so much. So, and our association also goes long way back. I was telling him yesterday when we were talking, my first visit abroad was to Japan for visiting us at Shimadzu facility in 93, 94, something like that. But anyway, it's a very pleasant morning here, and really appreciate Shimadzu and Spinco for inviting. And I'm going to talk briefly about, I understand that, no disrespect to food and the other industry colleagues here, but since I come from, my domain knowledge is in pharma, I may touch upon pharma a little bit. But it is very interesting to know that, pharma is very late to catch up on the mass, tool, whereas food and spices and those industries have been using it for zillions of years. So it's great to, know that. I think we have come a long way. If I go back to 35 plus years when I was a bench top analyst in a pharmaceutical company, we were probably somewhere in between TLC and the traditional color of solution, impurity methods and stuff like that. So in somewhere after I joined and a year afterwards or something, we got the first HPLC came. It was treated like a deity, It's in a room of its own, and then all small, chemists like us have to only see from the outside and revere and walk around. And the guy who can enter there and operate them was like, we used to kind of touch him, and to see that, how wonderful it is to see an LC in the lab. And from that to seeing, MS as a QC tool, it took almost 25, 30 years, While it was there sporadically, this instrument is used, but truly as a QC tool, and thanks to nitrosamine and other, knowledge that what we gained. What we are trying to say is technology, took a while for us to get there, but the way we are seeing the presentations today, and I'm sure that, it is accelerating even further. That is why I think, that our title for today is very important for pharma industry, is that it is no more a nice tool or a very fancy tool for QC. It is going to be a compulsive tool for QC in going forward. That's what I anticipate the MS technology to be. And mass spec, as we all know, has been an R&D tool in pharma and other industries too. And in a very, very sporadic manner, it is used in a QC environment. And as I said, that it has already been used in several other industries as a quality control tool. Now, when the advent of nitrosamine knowledge, I think, made the entire change, right? So until nitrosamine hit the pharma industry, there were very, very few applications where impurity methods needed beyond the traditional LC methods, right? So that also brings a lot of challenges and everything, and that's what I want to briefly touch upon before in this small delivery of notes. And just to give some numbers, how it rapidly ramped. If you look at the FDA and European agency or WHO and regulatory websites or the relevant database, you will see that several methods, several impurities have been identified. More than 300 APIs at least are listed with nitrosamine impurity, requirement, control requirements. So that means that much number of loads of, analytical methods for a QC laboratory environment. So this is going to have a significant impact, and as going forward, so we will talk about it. MS, as an ICP-MS, has always been used in the laboratory for elemental analysis and everything. So that is a different, technology, and the controls and everything that is required is not in comparison to what we generally considered as organic impurities requirement, what we need to have for our product. So of course both are important, but the controls and levels are much different from a technology standpoint. The USP has come up with the chapter 2, that is informational chapter, that is more than 1,000, 1469. So that talks about nitrosamine impurities, 4 analytical procedures are there, and then you have both LC and GC methods. European Pharmacopoeia too have come up with analytical methods. Now, when these methods become more and more popular, and it is not that the pharmacopoeia is discovering these methods, right? It is discovered or it is developed mostly by the industry. So that is why I was so wonderfully amazed by the amount of investment, time, energy, and the perseverance that yesterday evening we saw from Dr. Manohar, and that is not a surprise, knowing him. So the amount of efforts that as an instrument manufacturer or a service provider is putting, sometimes it is very easy to kind of see the presentation and just flip through and go through. But I think we need to pause back and see and what kind of partner we would like to have going forward as industry. If you are an industry leader or you are a decision maker, that what would be more sustainable, more scalable for you is service providers or partners who are willing to put their hands dirty and work for you, right? So I think I really commend Dr. Manohar and team for, I think it is a really selfless job. I mean, thank you so much for that. Wonderful, wonderful work. Now, it also brings a lot of challenges and considerations. Like one big challenge is a change in mindset, right? The QC environment is very, very black and white, and there are no gray areas, like you have go here and there. But QC is not really meant for interpretations. It is yes and no, right? So in mass spec, there is interpretation and things like that, data analysis. So not everyone is capable of doing it. So that is one challenge, obviously. But more than that, I think at this moment of time, the biggest challenge the industry faces is the lack of guidelines that or acceptance or a consensus on what kind of method transfers or method validation requirements are well set, method transfer requirement. Now what is happening is, if you have a method, you want to give it to a customer, you can't expect them to have the same analytical instrument model, make and all. Between the same organization, R&D to QC, maybe you could say, okay, I will have the same equipment here and there, so it makes it easy. But when you export this API or finished dosage forms to somebody else, obviously you can't dictate, okay, for this product, you buy this, model, and this product, you buy this model. It is not going to work like that, right? So I think there is a need for regulations to evolve and say, hey, this is not a real, the regular chromatographic technique where you say, hey, one method works for every instrument. So in case of mass, I think, it is necessary for regulations to evolve, the regulators to recognize what is really, efficient for the industry. So having every method to be revalidated just because it is a different analytical instrument model or make is going to drive the cost and efforts for the industry too. So I'm hopeful that there will be a lot of recognition about this issue, what industry faces, and make it easy for the industry by some smart solutions of how this method transfers or equivalencies can be proven, which is acceptable from a regulatory standpoint. Because science, we know that it is very easy to prove, but regulations are different sometimes. I'm not saying it in a negative manner, but I think it is the way it is built that way. So that I think is very important, and that is evolving, and I'm hoping that with more and more analytical methods being practiced in the pharmaceutical quality control, this will evolve quickly. Now, as I said, this is not going to be limited to nitrosamine or any specific impurities alone. I think that the all said and done, the routine, the conventional chromatography still lacks specificity, so as to say, right? So now when we have impurity methods that are not only quantifying wonderfully well, but also be very specific, especially with respect to unknown impurities and stuff like that. But I think it is only time that the regulators would kind of force for going for more specific methods like mass spectrophotometry methods for even routine analysis of other normal impurities, which probably doesn't have the same level of detection required, but still because of the relevance of that identifying unknown impurities. That is my anticipation, but it could take a while. But I think what is important for all the colleagues who are sitting here is to think about how do we develop our QC to fit into this new norm, which has already partially come true, that you already have 3, 4, 5, depending on the size of your product portfolio, already having instruments. So you need to develop that ecosystem within the QC, and that is what it is. And one more thing I want to talk about is that once this becomes a QC equipment, consideration for compliance becomes a little bit more challenging, in the sense like an R&D tool becomes a QC tool. So the elements that are built in an R&D tool is for certain reasons. But as I said, in a QC environment, we have strict boundaries, right? So how do we keep the strict boundaries intact, but we also have the flexibility of R&D in the same equipment? So, or the same ecosystem of that instrument, what we have in the QC environment. So that I think is also important. Plus, as I said, that the requirements for a chromatographic system today, if you walk into any laboratory, from a computer system validation or data integrity elements or audit trail elements, is significantly defined well and importantly captured. But when it comes to a development tool, a message or an error message or an action that is available in that tool may or may not be very look kindly in a QC environment, right? So it's very important to therefore distinguish. And my sincere request to instrument manufacturers like Shimadzu is to consider having a QC version for mass spec, especially with the operation conditions and the usage capabilities, etc. So that will become very easy to manage from a compliance standpoint for the QC environment. So I think, to summarize, I think that what is important to understand is that this technology is here to stay. And it is definitely going to grow further, not only going into, kind of how TLC methods were replaced by the LC methods. Probably our LC methods will be replaced by mass spec methods because of its affordability, the sensitivity that it brings, as well as the specificity that it builds. So there is a lot of work required between the industry regulations and also the manufacturers, service providers, to work collaboratively and determine the best ways to go forward and get our QCs, future ready in the sense that is going to come. And before I conclude, I also would like to, based on my observation walking yesterday in the laboratory, Dr. Manohar's laboratory. I think, I was telling them yesterday, it is very important to kind of utilize this as a national, a center of research, center of excellence, or such a wonderfully resourceful place, which the country need to kind of get benefit from. And I'm sure that Mr. Tyagarajan and team would be more than happy for providing this as a. I do not know really how it can be worked out, but it's very important to not to lose such wonderful work not being utilized by the fraternity of our nation's industry and regulators too, like Indian Pharmacopoeia or the Drug Controller India office or their setup, so that the regulators could easily utilize these capabilities to to understand the quality of medicines in this country and provide proper guidelines for patient safety, considerations. So, and also it is it would be wonderful or very apt to recognize the work Shimadzu and Spinco is doing for the industry here, because it's very easy to come and sell a product and be a nice smile. Anyway, they have naturally nice smiles, so a good homely environment, etc. But that's all done. Then every manufacturer and supplier would like to definitely keep that. But what really makes it different, and when I look at the efforts and the passion and the way that they would like to give it back to the industry by the science and the work they do, I mean, that's phenomenal. I mean, of course, the service is wonderful, everything, but what is important for me at least is that I realize that we should recognize the scientific work that has been put by the team for supporting the industry. So I wish them very well, and then I really appreciate for being invited in the august presence of the president and Mr. Tyagarajan and the younger generation here, and all my fellow colleagues and scientists from the industry, pharma as well as others, and wishing a wonderfully successful conference today and tomorrow. Thank you very much.